Tuberculosis 2022, FoVI-TB 2022. Antes de iniciar, me permito comunicar a todos los panelistas y asistentes que esta sesión está siendo grabada y transmitida por Zoom y simultáneamente por YouTube, Facebook y Canal Imagen TV UABC. Contamos con un canal de YouTube en idioma español que es Divulgaré SS UABC y otro canal de YouTube en idioma inglés que es Remi TV. La transmisión de este evento en los distintos canales se realiza con el apoyo del Centro de Estudios y Producción Audiovisual CEPA de la UABC, a quien le damos las gracias por su excelente trabajo. Este foro se está realizando gracias al apoyo de diferentes instituciones. La Escuela de Ciencias de la Salud, a través del programa de internacionalización en casa de la Universidad Autónoma de Baja California, a la Comisión de Salud Fronteriza México-Estados Unidos, la Red Temática Binacional de Salud Fronteriza, la Red Multidisciplinaria de Investigación en Tuberculosis REMI-TB, el Centro Nacional de Programas Preventivos y Control de Enfermedades, CENAPRESE, de la Secretaría de Salud del Gobierno de México, y la Red Nacional de Investigación para la Prevención y Control de la Tuberculosis. El Foro Virtual Internacional de Tuberculosis 2022 tiene como objetivo generar un canal de acceso a la innovación científica y la colaboración biomédica a nivel internacional y nacional con respecto a la prevención, diagnóstico y tratamiento de la tuberculosis, con la meta de cumplir con la estrategia de fin a la tuberculosis establecida por la Organización Mundial de la Salud. Esta estrategia busca reducir en un 90% el número de defunciones por tuberculosis y un 80% la tasa de incidencia de esa enfermedad para 2030, respecto de los valores de referencia que teníamos en 2015. La tuberculosis, desafortunadamente, sigue siendo una de las enfermedades infecciosas más letales a nivel global. Cada día mueren más de 4.100 personas de tuberculosis, lo que esto se traduce a 3 muertes por minuto a nivel mundial. Es importante recordar que la tuberculosis tiene cura si se detecta a tiempo y se completa el tratamiento. De igual manera, le recordamos a quienes nos acompañan en este foro que se emitirán constancias de asistencia. Les pedimos que revisen los requisitos para obtenerla en www.remitv.org. La fecha límite para solicitar la constancia se amplió al 26 de noviembre del 2022. Así también, para los que no se han registrado, en la misma página encontrarán el link para el formulario de registro, o bien por medio del código QR que estará apareciendo durante la sesión. El registro solo se realiza una vez. A continuación, les presento al doctor Ricardo Perea Jacobo, profesor de la Escuela de Ciencias de la Salud, UABC Ensenada, quien será el encargado de moderar la sesión del día de hoy. Adelante, doctor Perea.
Muchas gracias, médico José Luis. Me permito presentar al primer conferencista de esta octava sesión. El doctor Cuauhtémoc Licona-Cassani es profesor e investigador de tiempo completo, líder del Laboratorio de Genómica Industrial en la Escuela de Ingeniería y Ciencias del Tecnológico de Monterrey. El doctor Licona obtuvo su título de ingeniero biotecnólogo en el Instituto Politécnico Nacional, maestría en Ciencias Bioquímicas en la Universidad Nacional Autónoma de México y doctorado en Biología de Sistemas Microbianos en la Universidad de Queensland, Australia. El grupo de investigación del doctor Licona se enfoca en el desarrollo y aplicación de herramientas genómicas en microorganismos de interés biotecnológico y para la salud. Particularmente, su interés en el campo de la epidemiología genómica de Mycobacterium tuberculosis se centra en desarrollar herramientas bioinformáticas que ayuden a entender el genotipo de aislados drogo-resistentes. Su conferencia es FoVI-TB, una herramienta para la asociación funcional de variantes genéticas concurrentes en Mycobacterium tuberculosis según atributos clínicos. Doctor Licona, su plática tiene una duración de 40 a 45 minutos y posteriormente habrá una sesión de 10 a 15 minutos para preguntas. Adelante, doctor.
Muchas gracias. ¿Me confirman, por favor, si ven la pantalla?
Sí, doctor, se ve.
Excelente. Súper bien. Bueno, pues nada, muchas gracias a todos por la invitación, al comité que organiza estas excelentes charlas sobre tuberculosis. a los directivos de FoVI-TB y que organizan el Foro Internacional de Tuberculosis. La plática que voy a dar, que tengo preparada para esta ocasión, es una plática una actualización de los trabajos que hemos hecho sobre genómica de tuberculosis en mi laboratorio. Como bien comentan, pues está dedicado al desarrollo de herramientas de genómica funcional que permitan a los usuarios, en este caso la parte clínica, la parte médica, de los usuarios en general, usar y poder entender el fenómeno de drogoresistencia en tuberculosis, que es un problema que como bien sabemos, nos acecha y según como se ven los números y se ven las estadísticas, pues será un problema que si no se para por ahora, no hacemos algo ahora, se va a lamentar en unos 5 o 10 años, ¿no? Entonces, la plática que preparé, pues, refiere al desarrollo de la implementación de una herramienta y el uso de la herramienta en un set de genomas que hemos formado parte en su secuenciación de genomas autóctonos aquí de México, ¿no? De todo México. La plática, como bien comentan, pues se refiere a esta herramienta y bueno, vamos a empezar con la ahí está. Con la presentación. Lo que tengo preparado ahorita pues es una breve introducción. Realmente no voy a ahondar mucho, simplemente es pues que sepamos y tengamos en mente siempre que la tuberculosis pues es una enfermedad que ha estado con nosotros por muchos años, ¿no? Y esto, ¿qué significa para los microorganismos? Pues significa que a lo largo de...
We see that throughout thousands of generations, our bodies have adopted the capability of
características, ciertas atributos genéticos que le permiten vivir en el organismo, en el host, en este caso el humano, hablando de Mycobacterium en tuberculosis, pues por muchos años. Entonces es una relación muy antigua, y esta relación pues la vemos reflejada a nivel genómico, ¿no? A nivel de la organización, la codificación y el comportamiento de las proteínas que codifican los genes del microorganismo, ¿no? Eso significa la adaptación fisiológica al hospedero y evidentemente pues una convivencia.
So we see how the interaction between the disease and the human beings has evolved throughout the years. So we know the situation has become even more serious with the COVID-19 pandemic. So this is a very relevant disease, and obviously many strategies have been developed for decades. It's been over 100 years in order to try to stop the impact of this disease and the behavior of this disease. So the evolution of this disease is one that is very well known, and we can learn a lot from this. We have a vaccine that has been applied for over 50 years, and antibiotics, the first that were developed, obviously now they have effects, and we have observed some strains adapted to these drugs. So there's a lot to be learned still from this disease. So that's why we are here. That's what we work on. The deaths due to this the adaptation of these strains, in order to be able to live in a body that is receiving antibiotics. We know it's real. We see that every day. When we said almost a fourth of the deaths due to TB are because of multidrug-resistant conditions, because they have been able to adapt genetically to overcome these antibiotic attacks. So we know that the antibiotics that we use at the clinic, we all know, the first line antibiotics and some antibiotics that have been implemented the last 20 years, the second line antibiotics, just called. So not only that, but all the ways we use antibiotics since the 60s and today, we have had some efforts in order to, I don't know if I would call them efforts, but we have been trying to prevent the misuse or to fix this overuse of antibiotics. The fact is the same. There are different efforts in order to change this. So, especially since 2022, when the WHO updated they made a change in the use of antibiotics for TB, because we want to remedy the impacts of this misuse of antibiotics in the past. Another important case that is very relevant is how it is that we have studied and we have obtained the knowledge of the disease. Well, we have used the genetic variant analysis. I'm gonna make a pause right now because I'm gonna show you some slides on this later on. But speaking a little bit more about the efforts that have been made to combat TB, we know we're very familiar with this new action plan for the next 20 years or 15 years. That is the end of TB. So what we're trying to do is so the WHO is trying to come up with a strategy plan covering all the areas that are impacted by TB. But what we have to do on our side is on the epidemiology side of things. We are trying to reduce the impact of this that are very obvious, and we need to work on economic models, because we have been dealing with this disease for a long time. We see if we have a delay of 5 years on the achieving of the goals set by WHO, and if we don't compensate for this, we're going to have we're talking about the deaths of TB will double. So yes, and also the economic impact will be great. It's unmeasurable. So that's why it's so important to work right now. So one of the tools, one of the trends, if we remember the TB genome, it has been reported, it was reported over a decade ago. Since then, not only have we been learning a little bit more about the genetics of this pathogen, but we have had the last 5-6 years, maybe a little longer, of that we would call the democratization of the techniques, more advancements, some technological progresses that have allowed us to have this genome sequencing be applied in around the world, not only in privileged countries. In the case of Latin America, it's a very particular case where we all know the limitations that we have here for research and the lack of technology that we have in these regions compared to other regions around the world. So there are some advantages and some disadvantages to this. Some of the main advantages is that we can observe what the trends are that are leading the way of tracing the track for us, especially the application of genomes in the epidemiology. So we have here a good plan that we have, because we need to have efficient surveillance, and that's something that we have been doing here. We need to have good demographics. We need to overcome some barriers that we are still dealing with in order to extract the DNA in a more efficient way through primary samples, human samples, that are much more difficult to deal with in the culture in a lab. So in order to make it more time efficient. And once we get that, we need to have the genomic information. Then we need to know what to do, how to provide all this information to all the people in the clinics. We need to in order to inform them on vaccination, antibiotics, and such. So it's very important for us to see all the different cases, particularly in the lab. When I'm working right now in this lab, we are focused basically on the use of genomic information on TB. So here is where I would like to go into more further detail on how things are being done, how we were able to print out how we got all this information. So on the one hand, we have we could show an example of this genome. We have millions of the genes that make up MTB. So what we have done is we zoom into one of the regions of the genome. What we do is we use, since genome do not change much, since these are genomes, these are pathogens that are adapted, they have been adapting for many years in human body. So what we have done, what we have achieved throughout many years of hard work is the mapping of certain regions of this genome, certain areas, so certain targets, to be able to identify where these changes occur and how these changes are correlated to, in this case, to a geographic location. And also we can find some other points that are very important, that are fixed points that are correlated to certain behavior, physiological behaviors, such as drug resistance to antibiotics, for instance. And this way, what we do is we isolate in the clinic tens or hundreds of isolates. We generate isolates, and we sequence these isolates. And at the beginning, we thought it was not necessary to do this in the genome, and we only have to identify these points that have been very useful in order to classify the behavior of this microorganism. But what happens when we try to add some other attribute, or we are trying to see, as happening right now, we see a point in the human history where comorbidities such as diabetes are playing a major role, and public policies, because we see the incidence of diabetes has gone up greatly. So when these attributes, the new attributes or characteristics that can be, for instance, the origin of the isolate, whether the person has been vaccinated or not. So there are many more attributes that we would like to correlate with the genetic information we have available. Well, we sequence and we compare against a reference. As I'm using the pointer, you can see, you compare this against a reference, and then it's been built on a rational fashion with the purpose of covering more the more variability of genomes that could exist in the TB. But in a way, we can see that some disadvantages. So in this slide, I would like to show you where these possible discrepancies come from. So what we have done so far is we have sampled, and we got a sample genomic sequences, wherever we can do the most, there are more incidents or whatever, or is more economically feasible, and then we sample there and we generate a model. We set a model and said, okay, we take this target, this position, refers to a position that responds to the behavior of a drug-resistant situation, or maybe this one has correlated to this the genomic redistribution of whatever. So to generate these models, and we all use these models, and we use these platforms that are available in the literature to be able to say, okay, yes, we're gonna look at this genome, and this genome we put all the information on these databases, and then the platform will tell me what's the drug resistant profile and this genome, or what's the lineage of such genome in a very specified way. So when we ask our model about the variables, a direct correlation with the model we're doing here is what are the variables that they identify this isolate, because they come from the same regions, they come from the same model. The correlation is very good. So that's where we have built in a very good way. We have generated knowledge to get catalogs that year after year are published on strains or variables that refer to the behavior of drug-resistant phenotypes. On the right, you can see an image that maybe most of you know. That is when to describe the best way possible the distribution of a lineage in particular. What happens, however, when we start sequencing, or we want to sample, or we want to challenge the model in regions where we don't know if the behavior is the same or not. So let's say if I take the model and we have genome sequence and we don't know how applicable the knowledge we have generated by these models could be for genomes in some other parts of the world. So that's the case when we are right now, because unfortunately it applies to Latin America, particularly in North America, Mexico in particular, and Central America, and very few cases in Central America are representing this model. I don't know how useful this knowledge could be for people that are not well represented here. So we know that there are some hypotheses that could be generated, that when we start sequencing the genomes from other regions, as maybe there was a case that was published a couple of years ago in an African region, there are new lineages. So so we have to wait. The fact that we find new lineages, it means that they don't correlate these positions, the genetic positions, geographic positions, with what is expected. So we discover new lineages. So, and it's the case of this comes from a region of Mexican genomes, where we reported for the first time a strain of a sublineage, a lineage 4, X3 lineage. So where we can see there are some variables much more better adapted in order to against antibiotics. So we find drug-resistant phenotypes here in Mexico. This is one of the main things why we say, okay, we need, this one's blank. Okay, my bad. So there is a need to incorporate tools that will allow us to process genomic information that certainly is more abundant. And now we are in a time where there is more than ever, we have more genome information. So we need to incorporate efficient tools that allow us the identification of strains, of variants, and some phenotypical attributes and lineage. And what we suggest something I would like to tell you in today's presentation is the application of this tool. It's a tool that has to do with the efficient manipulation of the genetic information based on variants, and how we can subtract this functional information that could be used to generate some targets that could be incorporated into these models. So yeah, maybe just a reflection on how we can probably change the way analyses are being done instead of doing variant-based analysis. Maybe we can do pangenome genomes analysis. So when we say talking about this FoVI-TB tool, which is functional networks for TB genomes, what we are trying to do here is to have a double filter of attributes. First, we filter attributes. We see how the information is structured that is being analyzed on TB. So we have a correlation of the samples and the absence or presence of variants. When we have a variant and we have an absent variant, so we put that in those data metrics, which is in turn is related to a functional matrix, the epidemiological matrix. When we have all the information, all the different degrees of information, maybe it could be epidemiological data that we're all familiar with. So we come up with two matrices, and these matrices need to be filtered based on the several functions that those functions are set by the user. We should be able to say, okay, I'm gonna need to filter the data on this matrix is based on such drug-resistant attribute and so comorbidity attribute, and so I need to be able to see what are the representative data for this attribute after the first this filter of isolates. Then we need to come up with relevant information, all this packaged in such a way that the user can use this in a more efficient way. So going into further detail on how this tool is packaged. So the tool used is on the left top bottom, the left corner. See how the tool uses the structure of data that could be translated from what we get. We have the MTB-SEC, which is one of the most popular platforms, which is a matrix that correlates, as you well know, genes, chromosome positions, and presence or absence of all the different isolates. So based on this format, we generate a filter is a quality control methodology and functional control also, because in some data maybe we don't need to read or understand all the information. So the information that is filtered, some regions where they don't code, that is to say, this tool does not use some codes because it uses a functional purpose. So to remove intergenetic regions. We also filter, exclude, remove data. As you see, MTB-SEC use the reefs, the sequencing reefs, and then it has a quality control on the nutrients that could represent a low quality. And also how we can remove the background variations. And then with this, based on this metric, we come up with a data structure for each sample. For each one of the genes, we will have all the variations and the variable positions, and then all this information for each one of the samples is collected depending on the samples that the user decides to upload into the matrix. So this matrix will be used by the program to be able to structure the next matrix. So the user defines also what the matrix will be like. So it comes out with a simple table. What are the metadata? What's the metadata that included in the information? Out of all the information they may have based on their structure and their database. And from here, the user will be able to choose which attribute wants to filter. So based on this attribute, in this case, for instance, I don't know, so comorbidity or some certain antibiotic that wants to use as a pivot or as a filter. So the program will generate a prioritization of the data based on the value used given by the user. So they extract this data and they generate the matrix for the representative isolates for the first filter, which is the metadata. Based on this data, what we do, what the program does, it generates a visualization, and it does it based on networks. So for each one of the networks, what we do before that, we need to generate.
El usuario también escoge como un background. En el caso de una.
He's gone. I can't hear. He's back. So say if the user, for instance, needs to understand or visualize what are the genetic variations or the genes that contain variants, and see the genes that contain variants in a remaining uncommon position. So the problem will be the isolates that are resistant to rifampicin, for instance. So all the strains that are sensitive to rifampicin, and there will be the background.
A elección del usuario, removerse del resultado, ¿no? Ya con el resultado final de todos los genes que contienen variantes del subset o del atributo que
and depends on the subset or the attribute chosen by the user. So we come up with the statistics that for each one of these genes. Remember that we have an isolate group that were subject to this analysis for each one of these genes.
Frecuencia y de densidad, ¿no? Esto significa que cada
generate a position of losing here. Each gene will have a different value depending on how it was observed in another of the isolates in the other database.
Nosotros podemos hacer un ranking de cuáles son los genes, cuál es el grupo de genes que más se repite.
It's a group of genes that are most repeated based on the selected attributes. So now that we talk about how many of us are starting to use this tool, and the database that we use, we sequence 85 genomes of MTB in Mexico. And distribution of database could be seen here. So this is a raise in the incidence of
una distribución geográfica todavía baja con
of diabetes. And we also see the locations. So,
en 17 estados generalmente
basically the center and the north are more favored with this resistance profiles, and these genomes are isolated. We were isolated last year. We complemented the database with 80 genomes that were released by our work team in Veracruz, and we have about a database of about 80 genomes that are high quality genomes that have comparative information that we consider also to be good quality for drug resistant.
Ya nada más un poquito más de cómo es que se distribuyen. Este es un árbol de variantes, ¿no?
This is a variant tree where you can see distribution of the isolates. And obviously, as expected, we have a majority distribution of 90%, 95% of the genomes in our sample belong to lineage 4. So we have a good representation of lineage
autóctono, que es el X3, ¿no? Lo pueden ver aquí en la parte superior.
Endemic lineage. Sorry, keep losing him. And this tree we cannot see very well. we see transmission clusters that have been prior reported in Veracruz, for instance.
X3, ¿no? El linaje autóctono en México, pues se dice, se presume, es una hipótesis.
There's a hypothesis that need to be validated. We have observed that this lineage is related to drug-resistant phenomena. So that's what we're trying to generate or verify with our tool. So this is the distribution of a lineage in lineage 4 in our samples, and this is the distribution of the samples. This is very well represented in the center, and
la drug-resistencia así más o menos está en nuestras muestras.
In the northeast of the country. So everywhere the sample was randomized. So in most regions were sensitive, but we have some other cases of drug resistant that are profiled by the genome.
Hicimos para tratar de ver si los datos, la estructura de nuestra base de datos, pues tenía alguna valor o tenía algún valor.
So we, based on our database, we want to see if this database was useful for this new tool. So we generate
podemos generar a través de varias herramientas, ¿no?
Herramientas. Comparison and prediction. Sorry, keep breaking up. They're common. And also a very powerful tool which was released, and that includes the catalog, the release catalogs
por la Organización Mundial de la Salud, ¿no? Entonces,
of drug-resistant strains. By genotype, we have a good prediction, the solid as could be, based on the existing models, and the phenotype, we have the battery of assays that are done in the lab. And based on this, we generated a coefficient between the concordant. Before I show you the results of this study, we
las variantes, muchos de estos puntos que los modelos han determinado como
found that some relevant data points for the drug-resistant phenotype. And this is for some of the first
variantes que detectamos en nuestra base de datos. Aquí disculpa que esté.
Line the antibiotics. About the concordance obtained with this. This is not as expected based on the models, because if we model data in the same nature of genomes from different regions, definitely we expect higher, more active behavior, as reported in many of the publications that use TB profiler or MTB-sec. MTB-sec does not generate a drug-resistant prediction, but we use the algorithms of the variables that are different between TB profiler and MTB-sec, in order to verify these algorithms. These variant algorithms are variant validation. We compare them against the latest version. So basically, what we can see for each one of the drugs here, each one of the antibiotics, here we see in the concordance range, disagreement range. And we have phenotype against phenotype, and we said maybe those that can be improved, especially for ethambutol, streptomycin, rifampicin. The data that we wish were more agreement. So also in our database, it depends on the level of concentration level to which they were tested in the lab, each one of the drug-resistant phenotypes. But when we find a bunch of isolates, 10 isolates that were susceptible to all antibiotics, and that they were codified for kinetic variants. What we mean by this is determining for the drug-resistant phenotype, particularly in this distribution. 7, 1 for rifampicin, and this is a combination of antibiotics too. And the same way, we have 19 isolates that are very well representative, very good representations of the isolates that we have. It has had a phenotypical resistance to these antibiotics with this distribution, these characteristics, and they do not contain any canonic variant for drug resistance. So these are two icons where the models that we think, the models that are existing right now, they can be improved. And for that, we need to incorporate some tools that could use this information. So the first thing we did, if we have a model, a fourth of the data it does not match, is this in disagreement. So we use this data, and we only took the isolates, only the isolates that have a monoresistant phenotype. So these isolates that are monoresistant, we put them in the model, and we put them in the functional network trial, and then we took out all the sensitive variants, and we also removed, in order to clean this a little bit more, the genes that are subject to a discrepancy or disagreement in terms of the assembling quality. Many of the gene families, PPEs, and so some other those families were removed. So we generate these networks where we can see the genes that are shared, that includes variants, as H37RB, and then include variants that are, and each one of these variants, the genes are coded by the fitness score. The fitness score refers to how many, if these genes in that position, how many it accumulated, or if they are repeated in the same position, or the same position repeats in different isolates. So this is not to understand the genes that we see colored, the greener they are, the more representative they are, or more optimal for a trial, for research. So we have many canonic genomes of genes, but also find so many genes that could be good target of research. So on this side, theoretically, we see this. I cannot see this very well, but the outline of the genes of these nodes refer to the GO term. So this way we can see, I'm only going to show you the 10 genes that have the highest fitness score. Yes, the most repeated, they include most variants, and that they are going to agree in more than one of the isolates, from the range from 3 to 7 almost. So we have some genes that functionally may not say much, but some sense of DNA polymerase and some other that could probably start being part of some new research in order to try to incorporate or to see how prevalent these mutations are in other genomes or in their geographic, around the geographic region, and see if they are relevant to be incorporated. Also, what we did is, as we did a trial to do some monoresistant genes, and we also have this number of isolates, certainly is low. We are talking about 8 to 9 isolates, then they're only resistant to isoniazid and rifampicin, and only resistant to both, and that they are considered in the clinic as multi-drug resistant, but also only with rifampicin and isoniazid resistance. So we see genes that are among all the isolates that include these subgroups. These are genes that are shared, unique genes, and something that called our attention that we're having a hard time to interpreting is this. For isoniazid, we see many more genes that are shared with the multi-drug resistant phenome, and then we have 29, and we only have 3 genes that are shared between rifampicin resistance with a multi-drug resistant profile. So we can see, if we see in detail the genes, and we do much do more functional analysis in more detail, we think that it has to do basically with the activity of the antibiotic, that is simple, easier to generate resistance. Speaking of these two antibiotics, or we see there is a path in particular to generate this multi-drug resistance that have to be maybe first with rifampicin, insanazid. So we need to see maybe the order of the treatment, but we need to analyze still in the future with this analysis. So I think I'm running out of I'm running out of time, so I would like to say real quick that I would like to show you what the second way of looking at things. I mean, what I'm saying is, when you're talking about pangenomes, in our lab, we're sequencing genomes here in Mexico. And we need to do a pangenomic analysis. I think would be relevant, because we will be able to tell us going beyond just having to struggle and stop and make a comparison of the genome that we may have at a certain point in time against a reference. So we have to identify these variables that we know very well how they work, these spots. That what the pangenome does is, when we generate an assembling that is not subject to a reference, we have the possibility of finding genes or regions where that could be or are not present in the reference. When we do sampling of the genome versus a reference, these regions that did not find a match, they are discarded. So we need to do a new assembly. We will be able to identify some genetic regions that could be relevant and could be a participant or play a major role in the characteristics, depending on the geography or a much more local phenotype. What we have done so far, we start generating databases of genome databases, and then have a decent quality. Start to do pangenomic analysis designs to understand the genomes and the functions in pangenomics. What we do, we sample, we take notes, and we work on those functional notes. So what we see is a graph of 133 genomes. Out of 133 genomes, we have a cured database that we have in the lab, all the Mexican genomes. And we see in which proportion of genes, or in what proportion of the database, they share 100% of the genes, which proportion, what are the genes that are in this region. These are genes that, as you can see, they are not shared as much. And most some of the regions that are called the shell and the soft, when we find genes that share much less genomes. So by studying genes, maybe in this region, in this intermediate region, we see what are the functions that are there, how present these genes are, what's the prevalence of those genes, or these variants in those genes in these regions. So that might be able to help us know where these new targets are, how we can identify these genotypes that are identified in these variants that are least represented in the database. So I'm gonna wrap up with this. I just wanted to say, they have the tools that we have developed have helped us identify that some of the strains or isolates that we have, they disagree with the behavior. In some talking about the details, the rectification of the lineage Definitely, the classification of lineage differs from what we see in the lab and what is genetics through genomics. We have an incidence, a high incidence of X lineage in Mexico. We have strains, we have genomic material to study discrepancies, disagreements, and generate information on how they phenotypically behave, what would be other targets, what we might have in some disagreements on the isolates, and what these variables are, and which databases are. Maybe we need further research for that to work basically maybe in at least regional targets. So anyway, thank you for everything. We want to thank all the members of the people that work in our lab, Paulina and Axel. They are the major players in the development of these tools. Also, the different collaborators that may have made this possible. The sequencing have made the sequencing possible, specifically the Tech Monterrey, a tech-based section, and also people from the University of Veracruz, and in different research units and by the Mexican National Security Health System. We have they have been helping us with the DNA to be able to sequence those. Dr. Julio Castañeda, Dr. Antonio Enciso. And we also want to thank the financial support from CONACYT, a border project, Tecnológico de Monterrey, and Strain Biotech. Thank you for the logistics and the financial support for the sequencing. Without further ado, I'll just leave you my contact info, and if you have any questions, I'm here for you. Thank you. Thank you, Doctor. Thank you, Doctor, for your conference. And I'm gonna have a brief Q&A session on the different channels. We have some questions from the audience. The first one is, how can we foster the use of the complete genomes, considering the cost of the equipment and the staff? Because I think we have a team expert. Sorry, can't hear very well. What's the question again? How we can support this? Okay. Yes, well, I think that the best way to spread to spread of the benefits of sequencing a full genome is through this kind of events, this kind of talks. Is a very way to spread the word, going to different congresses. I don't think that using gene expert and using the kind of technologies, they are in a way they're efficient. That's why they are still there. They are part of the daily practices. But if we have the possibility of going beyond, well, we should try to generate not only knowledge, but we should also try to apply and develop new knowledge that is necessary. So I think it's very important to generate this comparison that will tell us, okay, the use of this technology help us improve in the screening of the isolates that we have to disagree, so that we can promote the use of these technologies. Also, I think it's important to highlight that sequencing, at least in my opinion, sequencing in Mexico, we cannot say that we are at levels where sequencing has been or could be widely used. So we need to invest and buy a sequencing machine, but sometimes it is not possible, because in order to lower the cost for sequencing, we need to sequence in high volumes. So in order to drop the costs, So that requires a high invoice for that. So maybe if we don't have this, what we could do is to sequence in facilities that are existing facilities. In Mexico, there are some, and there are many more in other countries. So you're talking about the WHO catalogs that Mexico is not very well represented in that number. So you think that has to do with what you mentioned, of the disagreements between phenotypic variants that are resistant, drug resistant, and those that did not have any drug resistant mutations? Well, the logic tells us that yes, they have something to do. To which degree? There are many factors that need to be taken into consideration. We need to be very careful and say, okay, this Mexican strains or the strains in our region are unique in the world. I think it's we need to be very careful. Take it with a grain of salt. Yes, the logic tells us that because it applies not only to TB but many pathogens that are sequenced. We know that there are new lineages, and these new lineages we also need to do good screening that is regarding time, and needs to be dynamic because maybe these lineages may change. So because we know the pathogens are much more fixed now with less incidence of genetic change position level, but it's a work that has to be done constantly. So we need to take this with a grain of salt, and I think there's enough evidence that will lead us to explore more. But we know that in our communications we have seen that there are some counter arguments that are very valid. That say it depends on a lot. If you see the strain is unmatching, so what's the concentration level that you used? So there is also this reluctancy to change with how many you do this, who did this, who studied that sample. So I think that the evidence that we have here is enough to say, yes, we have to prove this in 180 genomes, and now we need to double or triple the effort to have a better argument. And as for the pangenome, many of the genes are found in the core genome or in the accessory genes. How can it be analyzed if they're in two different locations? Well, the reality is that we are just starting to absorb many of the issues that come with handling this kind of data. So there is a limitation for us to be able to say a pangenome of 115,000 strains. We have a technical limitation. We're taking the baby steps towards the pangenomes of tens of hundreds of genomes. So I'm saying this because the data we have now they're very preliminary in this regard. So for the question in particular, in specific, I would say that canonic genomes are part of the core of the genome core. So maybe in some accessory genomes or soft, or maybe less represented, maybe find genomic functions that have to do their compensation functions or some other functions that might be relevant, but they could allow us to interpret why these phenotypes are not as and not so much agreement. So I think that's a way to follow, but from what we have seen so far, it's very preliminary. We still have to face many of the problems that have to do with data management at these volumes. We have never used a genome with 15, a set of genomes with 10,000 letters that are 4 million per 1,000. So this the volume is much more. and then we need much more capacity. So we are just starting there. Okay, great. So this software is already available. The software, yes. Yeah, it's available. It has not been published yet. We are about to publish it. Published in an article a few months ago. However, the software is available. It's available in a GitHub website in my lab. Or if maybe you can contact me. We might be able to send it to you, and so you can learn how to use it and help you throughout the process. Maybe right now it's not very user friendly, but whoever wants to use it, please be my guest. This is for everyone. Okay, thank you, Doctor. Thank you for a great presentation. We're gonna go to advertisement. This event's been sponsored by Banco with Matrix in Mexicali and offices in Ensenada and Guadalajara, and is also Vincent company with headquarters in Guadalajara, offices in Guadalajara, and distribution nationwide.
Ahora le damos la bienvenida al doctor.
And we want to welcome Dr. Sebastien Gagneux. He's a professor of infection biology and head of department of the Swiss Tropical and Public Health Institute by the University of Basel. After receiving his PhD from the University of Basel, he worked as a postdoctoral fellow at Stanford University at the Institute of Systems Biology in Seattle. Then he started his own laboratory in the MRC, National Institute for Medical Research in London. Before joining this Swiss TPH, his research focuses on ecology and evolution of Mycobacterium tuberculosis, with a particular focus on antimicrobial resistance. This conference is titled Ecology and Evolution of Multidrug-Resistant M2B. We want to welcome Dr. Sebastien Gagneux.
Thank you very much. Can you hear
Muchas gracias. ¿Me escuchan?
Oh, I'm the wrong channel. Because now I'm hearing the translation.
Ahora escucho la interpretación.
1 second. All right. So if I speak,
Muy bien. Si hablo ahora, ya no debo escuchar nada, ¿verdad?
Falta
¿Me escuchan
compartir.
bien ustedes?
Sí.
Bueno, voy a compartir y ya voy a comenzar.
Adelante, doctor.
Ok. Bueno, entonces, muchas gracias de verdad por la invitación.
It's really an interesting experience. Also, this dual language presentation. I think it's exciting. So we'll continue with drug-resistant tuberculosis, and I'm very grateful to the previous speaker because he obviously introduced the problem. And I will actually continue. Actually, I would start with a slightly provocative statement based on the latest WHO TB report. As you know, the WHO publishes a report on estimated cases every year. So this last one just came out a few weeks ago. And it's the first time since many years that the TB incidence has actually increased compared to the previous year. So we're talking about 2.6 million new TB cases that occurred in 2021. Now, when I refer to this provocative statement is the following. When you actually look at the number of multi-drug-resistant cases among these new TB cases, it's actually a rather small proportion. It's only 3% globally in average. I had a quick look at Mexico. If you look at the reported laboratory confirmed MDR TB cases for 2021, there were in fact only 382 compared to more than 20,000 overall reported TB cases, which is about 1.8%. So it's actually even lower in Mexico compared to the global average. Now, the reason I'm pointing out is because this observation, it turns out that this 3% of MDR has been rather constant over the last 10 years, which somehow comes as a surprise, knowing that TB has been treated with antibiotics for many, many years. So in theory, by now we should expect a much higher proportion of multi-drug-resistant tuberculosis, which, however, we don't really see if we just look at the global picture. And this, together with some other observations, really led to what I'm just referring to as the dogma, drug-resistant bacteria are less fit. Right? This idea that drug-susceptible strains, point to options, later options, that drug-susceptible strains lose their Darwinian fitness once they become drug-resistant. Now, this could be a loss in virulence and transmissibility, etc. And I'm saying dogma because some people believe that to be totally true. Others are maybe more flexible. We'd say maybe that's partially true, not always. I mean, biology, right, is often not just black and white, and we'll actually show you that this is actually a gray area. And one way to look at the gray area is actually by moving to a map, and that's one of the WHO reports 2 years ago where there was actually still a map. This year they didn't have that, which in this case shows the proportion of multi-drug-resistant TB among old TB cases by country. So the 3% I was referring to was overall across the whole world. If you do this by country, you actually see that the picture is rather heterogeneous. So particularly the dark red area of the world, right? They show actually a much higher proportion of multi-drug-resistant tuberculosis. And that's many countries of the former Soviet Union. The reason for this has been discussed a lot. I mean, we know that the quality of the TB control program plays a role. There's maybe other social factors which play a role we can perhaps discuss later on. But we thought there might be also bacterial factors involved. And so we were intrigued and actually to study this in more detail. For instance, you could think, we heard about the lineages, right, just before, and the sublineages, and we know these lineages are distributed differently. Maybe show a different map here, it doesn't really matter. The point is that we have these different variants and lineages around the world, which are kind of non-randomly distributed. So we have the blue variants or the Beijing strain, some of you might know, mainly in the east, in Asia. Then we have the red variants a bit everywhere. Then you have variants that are restricted to Africa. Point being that perhaps the distribution of these variants also have to do with this phenomenon of drug resistance. I'm going to go back to it quickly. I forgot to mention, so some of these regions of the world with very high MDR have been referred to as hotspots of multi-drug-resistant tuberculosis. So the question is, are there particular bacterial factors that drive these hotspots of multi-drug-resistant, together with potential other more social factors? So very long time ago, when I was actually doing a postdoc at Stanford, actually I tried to address this dogma experimentally by essentially generating resistant variants. So this is now rifampicin resistant. We heard about rifampicin is one of the key first-line drugs. So I generated different rifampicin mutants in 2 different strain backgrounds. These were susceptible clinical isolates, one from a lineage 2, one from a lineage 4 strain, in which I generated these rifampicin resistant mutants. And then I did competition experiments. Essentially, I competed, or I grew the resistant variants together with the susceptible variant, and essentially measured who was winning. And that's reflected here in terms of the relative fitness. So a relative fitness of 1, by definition, is the fitness or the growth, if you will, of the susceptible ancestor. And the bars indicate the relative fitness of the resistant one compared to the susceptible one. And just looking at across these different bars here, it really seems that the dogma is true, as far as rifampicin resistance and these different mutations are concerned. Because all of them have a statistically significantly reduced competitive advantage compared to susceptible strains. However, you can see that the bars differ. I mean, there's some that pay a dramatic cost here, compared to others which show a very small fitness defect compared to the susceptible strains. And there's this particular mutation here I would like you to remember. And for those of you working on resistant TB, you will remember this serine 450 leucine variant. The thing is that the nomenclature changed in the meantime, since this paper came out, and today we really talk about the 450 variant, which is equivalent. That this was used to be the E. coli nomenclature, as you might remember. So here's really the TB nomenclature, which is used today. So I'm going to talk more about this 450 variant later in my talk. Just remember that in this experiment, and actually others have done some similar work, we see that the fitness cost of this particular mutation in the lab is relatively small. It's not 0, but it's relatively small compared, for instance, to some of these other variants. Now, this was in the lab, and in lab strains, the question was, okay, is this just some weird lab artifact? And what about if we look in clinical strains? So this was the second part of that paper, where we actually took paired isolates. So this is an initial susceptible isolate from a patient who developed resistance over time to rifampicin. And then we had a follow-up isolate which was resistant, and did the same thing. We competed these 2 isolates that came from the same patient, and we had essentially 10 of these pairs. And what we can find is that, again, there was heterogeneity in terms of this fitness defect caused by the resistance mutations, consistent with the low cost of this particular variants here, the light gray bars here. We found actually some cases had 0 defect, whereas others still had a very strong defect. So there was a difference between some of the clinical strains compared to the lab strains I showed before. At the time, we didn't really know what was happening. But how could we explain this? We could only come up with a hypothesis, which is, so I'm showing the same graphic here I showed you before, but now the hypothesis coming along, which is that over time, because evolution doesn't stop at this stage, it continues, and so you can have so-called compensation, meaning additional mutations accumulate, which bring the fitness of these resistant strains back up. And at the time, that was a question mark, right? Does this happen in TB? We knew that this happens in other bacteria, even in some viruses, the question was, does something similar happen in tuberculosis? And to make a very long story short, publish it is the study is published several years ago again. I mean, just to summarize what we did. So we did experimental evolution. The idea is that you force essentially the TB strains to evolve in the lab in the hope that they might actually acquire the so-called compensable mutations. So we did this with these lab strains. And then what we also did, because we had access to these serialized lists, the same as I just referred to, we then sequenced and looked for additional mutations. So not just the resistance mutation, but potential additional compensable mutation. So putting this together, again, so that long story so short, published paper, yes, so we did find compensable mutations in the RNA polymerase. So that's actually important. So let me explain. This is a diagram of the RNA polymerase structure. RPOB, that's the red subunit, because this polymerase is comprises various subunits. So the RPOB, the beta subunits, that's where the resistance conferring mutations occur. So the classical canonical rifampicin resistant mutations, which have been known for a long time. What we found in addition are new mutations in these other subunits, which always co-occurred with a resistance mutation and really were associated with an improved growth of these isolates in the laboratory. Then the problem was, of course, at the time we published the paper, we were criticized in the sense that, again, you're showing this is in the laboratory, who really cares? What about the real world? Some people even said, you should do these tests in mice to show that they really do what you think they're doing. But then we said, okay, let's skip the mice, because at the end of the day, humans are not mice, and mice are not humans. What we really care about is what happens in humans in terms of transmission, and which is the ultimate measure of fitness, right? Is how well do these MDR strains transmit from one patient to the next. And so for this, we actually went to one of these hotspots of multidrug-resistant TB, which is the country of Georgia, which is a small country. It's not quite as dark red as some of the neighboring countries, but nevertheless, it has quite a lot of multidrug-resistant TB. I must say now less than it used to have when we started the project. Fortunately, we started there about 10 years ago. And the study I'm going to show you is really a 6-year study, which includes essentially all available MDR and XDR TB cases from 2011 to 2016. And we managed to recover about 70% of these. And that's a nationwide study. That was the advantage of working in a small country, which we really wanted to do. So to be able to look at this nationwide and population-based. And so the first thing we did with these TB isolates was to genome sequence. And this we did in collaboration with the National Center for TB and Lung Diseases at Tbilisi, which is really the reference, TB reference lab in the country of Georgia, which facilitated access to these strains. And then we have Sebastien and Chloe, who were previous PhD students who did the data analysis. And so here's a phylogenetic tree of a bit more than 1,600 MDR/XDR genomes from Georgia across these 6 years. What you can see is that there's an overrepresentation of lineage 2. So lineage 2 / also known as Beijing is really responsible for the large proportion of these MDR strains. This was somehow expected. We knew this from other previous studies from Georgia, but also from neighboring countries, that MDR strains in these areas of the world are primarily driven by lineage 2 strains. What what was new, though, is when we looked at the proportion of these that carried the compensation. In other words, a mutation in RPOA, B or C, which are compensating. So it's almost 70%. So a very large proportion, which already indicated, okay, at least they occur in clinical strains, and if they are there, presumably they're doing something. We knew they're doing something in the lab. The question was, of course, now, what do they do in the field? And the other thing is, because this was a nationwide study, we also had access to prison isolates. So from TB patients in prisons, and this is going to be important in a minute. So what we did next, we actually used tools which combine this genomic information with the date of isolation. And this allows us to reconstruct transmission trees on the one hand, and also calculate so-called secondary case numbers. In other words, to find out how many people a particular TB patient infects by reconstructing the directionality also of the infection. So essentially, who infects whom. And we use this as a measure of transmission success. And what was key here was what was actually important. We had to, of course, also control for other factors, because we know there's many host factors and others that might also influence the transmission success of TB strains, such as sex, age, previous TB, etc. And so we performed a so-called multivariable analysis, which actually adjusts for these potential confounding effects. And what I'm going to show you here is now the three key variables that we found associated with an increased number of secondary cases. And we really essentially found what we hoped to find, which was compensatory mutations. So compensatory mutations independently really contribute to a larger number of secondary cases if patients infected actually are infected with strains carrying compensatory mutations. Remember here, we're only talking about multidrug-resistant strains, right? It's comparing multidrug-resistant strains with and without compensatory mutations. We're not talking about susceptible strains yet. This is in the second part of the presentation. We also found that lineage 2 was associated with a higher number of secondary cases among MDR strains. So this is perhaps not surprising, given also the overrepresentation of this lineage in MDR strains in that region of the world. And then something we did not have on the radar, but in retrospect made sense and perhaps even serves as a positive control, incarceration. I mean, we know prisons are extremely conducive to TB transmission, particularly in conditions as they used to be in Georgia and many former Soviet countries, which fortunately in Georgia, this has improved a lot. Over the last 10 or so years, or even 12 years, at the time, this was actually much worse. So, but at the same time, this raised a new hypothesis in the sense that we said, okay, we knew that prisons, or we know that prisons play an important role in TB transmission. However, what we then thought, perhaps it's not just TB transmission that prisons help, if you will, or support, but maybe it also drives evolution of the TB strains themselves. In other words, is it possible that prisons serve some kind of as breeding grounds for particularly transmissible MDR strains? Now, again, it's MDR because that's what we're focusing on here. And there seems to be some support for this idea, because when we look at incarcerated as opposed to non-incarcerated isolates, patients' isolates, there was an association between compensation and incarceration. So in other words, the compensatory mutations were overrepresented in prison isolates. And that led to yet another idea or notion or hypothesis, which is that perhaps these highly compensated transmissible strains were generated or emerged in these prisons and gradually are released into the general population. And one way to try to look at this was by, again, looking at these transmission trees. And I'm showing here one of the largest one, actually the largest transmission tree that involved a lot of patients over the 6 years. Let me explain what this shows. So every dot here or circle is a genotype and a patient. The color coding is purple for prison, orange for civilians or general population. The darker the color means the younger or the most recent diagnosis date or isolation date on the asthma. And the arrows are the inferred directionality of transmission. If you see if the whole thing starts around here, there's a tendency to go from light purple to dark orange, indicating that perhaps what we hypothesize is actually true, that is a trend for these MDR strains to move from the prisons into the general population. And in fact, if we then extrapolate this or do the same analysis for all transmission trees in the population that we looked at, we see a clear trend going from mainly prisons into mainly civilians over the 6-year study period. And then you can actually extrapolate this to all MDR strains that have been reported in the country of Georgia over the 6 years. And if we do this, we infer that almost a third of all MDR-TB cases in Georgia can be either directly linked or indirectly linked through transmission out of prisons, indicating that prisons really, at least in Georgia and around that time, played an important role in the spread of multidrug-resistant tuberculosis. And this also indicates that making sure that TB patients coming out of prisons actually treated properly and followed up and not lost into the general population without follow-up should be a priority to prevent, presumably not just MDR-TB, but TB in general. Here we just illustrate this with MDR-TB in particular. Now, moving to the second part, I already alluded to this briefly, is that, of course, you might then say, fine, you looked at just drug-resistant strains. It seems that compensation really plays a role in terms of improving the transmission of MDR strains compared to non-compensated MDR strains. But the true question might be, so what about susceptible strains? How do these MDR strains, compete against susceptible strains? So for this, we essentially extended the work in Georgia by including drug-susceptible strains. In this case, we only had 3 years worth, because this was now a prospective study where we essentially collected prospectively all strains, not just the resistant ones, but also the susceptible ones. So we got a much higher coverage, almost 90% across these two types of strains. And then we joined forces with Tanja Stauder's group, who at ETH in Zurich, who is really at the forefront of developing new so-called phylogenetic models, which are even more sophisticated than what I just mentioned earlier on, to really measure, quantify the transmission of pathogens. This has been used a lot in the COVID epidemic, but it's used a lot for many other viruses. And Ethel, a postdoc in her group, worked together with Chloe to analyze this data using this novel method. So it's the same principle. You start off with genome sequences, and you add also the date of isolation, and these methods would infer most likely success transmission. In fact, 2 different measures I'm going to mention in a minute. So we have the MDR strains on the one hand. That is a smaller data set now, right? Because it's only 3 years. It's not 6 years. But then we add the susceptible strains, which were almost 3,000. So that's a large amount of data. What you can appreciate here immediately is the lineage distribution. I mean, we see almost a mirror image amongst the susceptibles. We see much more lineage 4 compared to lineage 2. And this is highly statistically significant, this difference, right, in lineage distribution. Again, not necessarily surprising, but at this scale is not necessarily what we're expecting. So a very strong effect. So clearly, MDR in the country of Georgia is primarily driven by lineage 2. And the question comes, so why is that? So what is special about lineage 2 MDR strains that make them so successful compared to the susceptible strains generally? And so we came up with a hypothesis, and maybe some of you who have been following the literature on Beijing strains and the association with multi-drug resistance, which is not new. Again, I mentioned there have been multiple hypotheses, higher mutation rate, etc. So more likely to acquire resistance. What we're proposing here is something a bit different. What we're proposing here is that these particular lineage 2 strains from Georgia, and have to point out, that's not any lineage 2 strain. It's really the one which circulate there. They might be just particularly well adapted to cope with the fitness cost of resistance. And that's what we set out to test with these phylogenetic models together with Tanya Stadel's group. And so this is now the first part of the result. Of the second part, I need to explain this a bit. So what these methods do, they actually estimate 2 different measures of transmission success. One is the transmission rate per unit of time, and this gives you actual distribution. And then you have the effective reproductive number, which also incorporates or estimates the duration of infectivity, or how long a patient remains infectious and is able to infect other people. And turns out that this difference in here is actually not that important, because we essentially see the same picture. So what we see is, on the one hand, we have the drug susceptible transmission success in light gray, and in blue we have MDR. What you can see in lineage 2, in both of these measures, these distributions overlap, meaning that essentially there's no difference in transmission success when we look at the MDR in lineage 2 compared to the susceptible counterpart. Interestingly, in lineage 4, there's actually a very big difference. So the transmission success of MDR in lineage 4 strains is actually much less compared to susceptible strains. And this seems to support this hypothesis that for some reason, lineage 2 MDR strains from Georgia don't suffer a fitness cost in terms of transmission, whereas lineage 4 strains, MDR strains, do. The next question was, okay, so what is special about this lineage 2? Is there something else we can do? And yes, and now remember the mutation I was referring to early on in the lab, right? The RPOB SV450L mutation, which in the lab already was associated with not a 0, but a small fitness cost. So we said, can we look at this here using transmission as opposed to with lab growth? Do we see a similar effect by doing a stratified analysis? Now we have 3 peaks. Again, we have susceptible as a reference in gray. We have now the red peak here is MDR strains that carry the RPOB 450 mutation, which, as you see, have actually a small difference in lineage 2, again, in both measures. And we have other MDR strains that carry other RPOB mutations, which we know, again, in the laboratory, are associated with a higher fitness defect. So it seems that in lineage 2, we see the measures in the lab essentially mirrored. Whereas in lineage 4, again, things that are different. There we see that irrespective of the RPOB mutation, the fitness defect is always there. So what this indicates now is that we have now an interaction, a genetic interaction between this particular mutation and what we refer to as a strain background. But we know lineage 2 strains differ from lineage 4 strain by at least 500, maybe 1,000 mutations, SNPs. And that means that if you have the exact same mutation, this is influenced. The effect of this particular mutation is influenced, and this is really a matter which is referred to as epistasis, right? The change in phenotype inferred or induced by one particular mutation when it interacts with other mutations. In this case, with actually very many mutations which are defining of a particular strain background. Now you might say, okay, so what about compensation? We clearly see there is an effect of mutation. And they say, okay, fine, so we can continue, we can further stratify, right? And now it becomes a bit more complicated even. Now we have 4 peaks. What we did here is really to say, okay, we see there's an effect of 450. Do we still see an effect of compensation? Or maybe compensation doesn't play a role at all, and it's all explained by the mutation, the 450 mutation. Turns out, no, that's not the case. So here now we're looking at the brown distribution, which is MDR strains that have the RTOB mutation and also have a compensatory mutation. These have actually a high fitness, even higher than susceptibles in this case. Here, essentially no difference to the susceptible strains in this particular measure. Whereas MDR strains with 450, but without compensation, are a bit less. And then the blue variant are the ones without 450 and without compensation, which are the least fit. Again, this effect we see in lineage 2, but not in lineage 4. So for some reason, lineage 4, we don't see a signal of compensation. So taking all of this together, we have a model, which I quickly would like to illustrate here. So it's this, what we refer to as 3-way epistasis, because on the one hand, we have the strain background, as I mentioned, right? A particular lineage. Now, in this case, it's lineage 2, which jumps out as being interesting. Then we have a resistance mutation. Of course, in reality, it's more complicated because we have many more resistance mutations as these strains are resistant to many other drugs. In this case, it's the 450 mutation, which somehow jumps out as being interesting. And we have the compensatory mutations, which all interact in conjunction and together influence the transmission fitness of a particular MDR strains. And that's at least what we see in Georgia. And in terms of conclusion, now linking it back to very early, my point about the hotspots. So what could be driving the emergence and also perhaps maintenance of hotspots of resistance? Well, what we think is really going on, we have a combination of a conducive environment, which, for instance, could be mass incarceration, as it used to be the case, and perhaps even some cases of the Soviet republics still have this situation. As I mentioned, Georgia improved a lot in the meantime. So an extremely conducive environment with poor TB control, massive crowding, alcohol, drugs, you name it. Then you perhaps by chance, right, a lineage 2 strain makes its way. You let it evolve in absence of good treatment or a lot of poor treatment, so resistance emerges. It will have a particularly low fitness cost mutation, which is 450. It further evolves, acquires compensatory mutations, and then you have this phenomenon of epistasis, which together would then lead to a hotspot of resistance. Clearly, in reality, it will not be that simple, but again, this would be our model to think. And because I do still have some time, I think. Yes, I'm trying to see. Yes, so So I thought, okay, there's some other unpublished data I wanted to share with you, because we tried to, almost a bit in parallel with some of the work in Georgia, we said, you know, if any of this is true, we should be able to also replicate some of these findings in a different place. And so here we move into South Africa. And as you, South Africa is unfortunately known for a lot of TB, and particularly also drug-resistant TB. You will note that South Africa is not as dramatic, right, as some of these other areas, but in terms of absolute numbers, still, it has a lot of, obviously a lot of TB, but also quite a lot of drug-resistant TB. And that's particularly true if we go to the place we went to, which is Khayelitsha, which is one of the neighborhoods in Cape Town, which it's not a prison, right, in the classical sense, but you could almost, you know, interpret it in terms of some kind of prison, in terms of the effect of crowding, right? And this work we did in collaboration with Helen Cox from the University of Cape Town and Rob Warren from the Stellenbosch University. And here we have Gallo, who is a postdoc who actually did most of the analysis of this part. And we did a very similar analysis. In this case, the advantage was we had actually even a longer time period to look at. The disadvantage was that we didn't have the susceptible strains. We only had the resistance, let's say the rifampicin resistant strains. So there were at least resistant rifampicin. Turns out there's quite a lot of rifampicin monoresistance in this area, but that's a separate talk. Again, the point of this work here was to see whether we could find similar phenomena that we had seen in Georgia. And the answer is essentially yes. So what is shown here? So Gallo was using a slightly different method, which is known as TransPhylo, which does a similar thing. So it doesn't quantify the number of transmission events per person in this case, but it tells you whether someone has transmitted TB or not. And that's indicated here. Now we have three categories along the lines of what I showed before. Again, the 450 mutation, remember, right? So we have the non-450 carrying strains, then we have the 450 carrying strains, and then we have the 450 with compensation. And you can see there's a general trend, right, of having more transmitting patients depending on which type of mutations they carry. Now, of course, again, it was very important to do the so-called multivariable analysis, which we did in the first paper also. And without going to the details, because it's a bit crowded, I'm just going to highlight what we find. So we do find, even if we control for all of these other potential factors here, which might influence TB transmission, we do find that patients were infected with the MDR strains or the rifampicin-resistant strain, which carries this RPOB mutation, and the compensation mutations are indeed more likely to be transmitting multidrug-resistant TB. So somehow adding or confirming to some extent our findings from Georgia. Now, Gallo had another idea, because he said, okay, there's something else I could look at, which is the number of other resistance mutations that these strains carry, because they tend to carry many additional resistances. They're not just resistant for rifampicin, isoniazid, streptomycin, etc. And what he found is that there was actually also an interesting effect, it's a correlation, right? An association between the number of additional resistance mutation depending on these three categories I just was referring to. And he found this when he looked at all isolates, right? So non-450, 450, 450 with compensation. So 450 with compensation had more, significantly more in average resistance mutations than the other two categories. And because this could be confounded by multiple isolates per cluster, he did the same analysis using only, which is much more stringent, using only one isolate per cluster, but this remains statistically significant. So this suggested something else a little bit. I must say also this observation was shown before. Others have shown that compensatory mutations are associated with more resistance mutation. So Gallo confirmed this, but this raised another idea, yet another hypothesis, because here we're talking about resistance amplification. And presumably, at least partially, resistance amplification, so gaining additional resistance, happens inside patients, right, who receive drugs. So suggesting that perhaps these compensatory mutations, not only do they help the transmission of TB from one patient to the next, but they might also increase the within-host success. In other words, the within-host fitness of or competition of TB inside a particular patient. And therefore, patients with these compensatory mutations are more likely to acquire additional resistance mutations. And one way he tried to look at this was to use a very rough, and we realize this is really extremely rough, proxy for within host bacterial replication, which is smear positivity. And presumably, we know this is imperfect, right? But still, it's just one way to try to look at this. The idea is that smear positivity will be correlated with the amount of bacteria that live in the lung of these particular patients in average. We know in reality it's not that simple. But nevertheless, when he did that, again, just looking at univariate, which is actually shown here, but more importantly, multivariable, it actually did, in fact, see that, again, patients infected with strains carrying 450 and compensation were, in average, more likely to be smear positive compared to patients infected with other strains, indicating that perhaps something is going on also not just between patients in terms of transmission success, but also within individual patients. And with this, I'm going to come to the conclusions of the second part. So compensatory evolution affects both. We think it does affect both in terms of, on the one hand, between patient fitness, which essentially is transmission. And we have support for this from Georgia as well as from South Africa. And new now in South Africa, indicating that perhaps within patient fitness is also improved by compensatory mutation, and giving rise to additional resistance mutations, in other words, drug resistance amplification. And with this, I would like to end, thanking other collaborators I haven't mentioned, funding agencies, the other members of the lab, and thank you very much for your attention and the invitation to present.
Muchas gracias, Dr. Gagneux, por su interesante conferencia.
Thank you, Doctor, for your interesting presentation. Now we have a Q&A session. We have some questions for you. The first one would be, why in the dogma you expected to see fitness lower in resistant bacteria?
So I mentioned, what I didn't mention is that there were actually early studies in the 50s at a time where obviously there was no molecular biology, but what people were doing at the time, they were putting TB strains into guinea pigs. And there were studies where this was done, where the virulence of these strains in guinea pigs was comparing susceptible strains, and in that case, it was isoniazid resistant strains. And turned out that many isoniazid resistant strains indeed were attenuated. They showed a reduced virulence in guinea pigs compared to fully susceptible strains. That was one indication. But the other is, and that's a more general feature, just explaining the fact that antibiotics tend to target key enzymes, which tend to be highly conserved. So these key enzymes do important things in metabolism of these bacteria. And they have evolved to be as perfect as they can over many millions or thousands, millions of years. What happens with resistance, right? In the case of rifampicin, for instance, you have a mutation in this optimally evolved RNA polymerase, and the bacteria is facing a trade-off in the sense that either it's killed by the antibiotic, or it mutates away from this perfect evolved form of the RNA polymerase. And by doing so, it pays a price, right? Because it's not as optimal as it used to be. But at least it's not killed by the antibiotic, right? So, but compared to the susceptible ideal version, it has a reduction in fitness, in generally speaking. And the same idea, right, can be applied to all antibiotics to some extent, right? As long as we're talking about the highly conserved targets. So that's a bit the rationale, if you will, of the dogma. And the truth is, it's true. I mean, if you remember the first experiment I showed you, I mean, essentially all the resistant variant had a fitness defect compared to susceptible strains in the lab. It's just that in reality, it's more complicated because then additional things happen, which is essentially compensation.
Thank you, Doctor. The next question is, what kind of control do you need to be established in order to prevent prisons from becoming hotspots?
For prisons to becoming hotspots. So that's extremely important question, and I haven't mentioned that, and that's also the reason the situation changed and improved much in Georgia. Essentially screening, right? So you need to actively look for TB in prisons. And ideally you do that at the beginning when the inmates come into the prison. You want to exclude that they have TB as a start. Then you need to actively follow them up inside the prison, making sure that you discover potential TB cases and treat them immediately, properly. And importantly, when they leave the prison, even if they're not completely cured, right, which is obviously possible, MDR patients might have to be treated for a longer time, you have to make sure that you follow them up. Just not forget about them and they disappear into the community. And that, I think that's what happened in the past. And I think generally TB in prison is underappreciated. I think that's true for many parts of the world.
Thank you, Doctor. Your next question. Do you have an estimate how many contacts we need for a compensation mutation to appear?
And how many what?
How many contacts there have to be in order to have this? Do you have an estimate of the contacts? How many people are necessary for a compensatory mutation to appear?
I think I have no idea. I mean, again, presumably these compensatory mutations emerge inside individual patients, right? But we don't really know. What we speculate is that it might take some time. And the reason I'm saying this is there are other places that are not quite as bad. When we look at the red map, or the MDR map, there are these red areas. And the truth is that most of the places in the world, they don't have a huge problem with multidrug-resistant tuberculosis for the reason I said at the beginning. And that was a bit of a surprising fact, right? Remember these 3%. Even in Mexico, it's actually even lower than 3%. So, I mean, you can only speculate that presumably you need to have a bad TB control program for quite a long time. And then maybe other things, like for instance, prisons and perhaps again, speculation. Perhaps you have HIV or extreme crowding, as we see in Khayelitsha in South Africa and some other places. Are the necessary ingredients for these changes to happen, right? But I really don't know the answer to the question.
Yeah, hay que investigar.
Yeah, we need more research on that. Okay, so the compensatory mutations in lineage 2, can they be considered for new pharmacological targets?
I think it's an interesting idea. As I mentioned, the compensatory mutations that we do know, and there might be many more that we don't know, particularly also in relation to other drugs, right? We very much concentrated on rifampicin. And all the ones that we do know are in the RNA polymerase. And as you know, RNA polymerase per se is already the target of rifampicin. Turns out it's targeting the beta subunit. The compensatory mutations occur in other subunits, but also in the beta subunit. I forgot to mention that this was shown also by others. So you can have rifampicin-resistant strains that carry more than one mutation in the beta subunit. Whether this will be potential new targets of drugs, perhaps, I don't know. It's an interesting idea.
Gracias. La siguiente pregunta, el comportamiento.
Okay, the next question, the behavior observed in prisons could be replicated in immigrant populations that live in shelters?
Yes, I think I also have to be careful, right? When you say behavior of immigrants in shelters, it's not really the behavior, it's the living conditions, right? I wouldn't want to blame the inmates, right? It's not their fault, right? They have to live in these conditions. So I think we need to turn the blame around. Is the people who build and conceive these shelters that they need to make sure that this cannot happen. It's not the immigrants themselves, right? To make that very, very clear. But yeah, in principle, yes. I mean, if you have a catastrophic shelter where you're putting hundreds of people under extremely poor condition, you don't control, you don't. This actually already sounds like very much like a prison to me. So in theory, yes, you could have the same problem.
Muchas gracias, Dr. Gagneux, por su interesante.
Thank you, Dr. Gagneux, for your interesting presentation, for your time answering these questions. Thank you so much.
Thank you for the invitation.
Muy bien, pasamos a los anuncios.
We move on to the announcements. To all the attendees that are interested in getting an attendance certificate, the deadline for this is next November 26th. The requirements for this certificate can be found in www.remitv.org. We want to thank our sponsors, VAMCO and ISA, also the interpreter Claudia Valencia, who interpreted both presentations, English into Spanish and Spanish into English. The English conference can be found in RemitTV YouTube channel. We also would like to thank all the support for the streaming of this session to the Audiovisual Production Studies Center by UBC Mexicali. We want to thank you for being with us throughout this forum in all its 8 sessions. And we also want to invite you to join us next year. We also want to invite you to follow us in our social network on YouTube, RemitTV, YouTube Divulgare, ECS UABC, Facebook, and Instagram under RemitTV. Thank you. Have a wonderful day.